Elisa, sono parte dei miei volontari. Volevo sapere se hai questioni, domande,
non c'è formalmente un debito, ma se
noi saremo live e quindi è importante che loro abbiano il microfono quando fanno la
Okay, good afternoon, everybody. Welcome to the lunch seminar. Please take a seat. We allowed a few minutes delay. Thanks for convening at the talk by Professor Chiara Cirelli from the University of Wisconsin, talking about sleep, a key topic in the context of time, as because you're all aware of, a good percentage of our lifetime is spent sleeping. And what is happening during sleep is enormously important, as we will learn, of course, from Professor Cirelli's talk. There are a number of parameters that influence our health, but also disease, but also social aspects that impact time and sleep and the quality of it, and how the reflections on the quality of our life in general, including private life, of course. So Chiara was born in Ferrara, in northern Italy, did her studies in University of Pisa, where she basically started all her career in neuroscience. Continues very successfully in the University of San Diego, where he started basically the hardcore studies in sleep biology, and then got a position at the University of Wisconsin since a couple of decades now, where he's a full professor in the university there. And she's one of the leaders in the field of fundamental biology. And she's been using a variety of model systems, including the Drosophila, to perform large-scale genomic studies to understand which networks underlie the regulation of sleep, then moving also to other organisms, including the mouse and so on, to go deeper into this. Among many other things, also she came up with seminal theories about the importance of synaptic plasticity in regulating the on-off, state of our sleep-wake cycle, the on-off state of contacts between neuronal cells, basically, that switch off the mode and start a different life in a way, or recharge basically their batteries in order to perform during the awake time. With no further ado, Chiara, please join me on the podium, and everybody may be welcoming Chiara to your house. Thank you very much for being here. Looking forward to your talk.
Thank you very much for the nice introduction and for the opportunity to be here today. It's my first time at KAUST and first time in Saudi Arabia, so I'm very, very grateful for this opportunity. It's been a fantastic experience for me. So my task today is indeed to tell you a little bit about what's happening in the brain when we sleep, and most importantly, why we sleep. And that's the riddle that we have been trying to solve over the last 30 years or so, as Professor Lando mentioned, is a long, enduring battle, and hopefully we will have the answer. But I'm going to provide you with some cues that we have about why sleep is so important. And in the context of this program about time, it's indeed very important to remind everybody that indeed we spend more or less a third of our time asleep across the entire lifespan. This is a survey that was done a few years ago in the United States. People report spending 36% of their hours asleep. But it's very consistent finding across different societies and cultures. More or less a third of our time is spent asleep. And of course, you think about kids and newborns and babies, they sleep a lot. But in reality, we do sleep a lot also in more mature age. So this is a description across development of the total amount of sleep on average at the population level. And the two darker colors that you see are the light orange is the so-called non-REM sleep, which is the bulk of sleep, 70–80% of sleep in an adult. And the darker color is REM sleep. And what you see from left to right is that from birth until very old age, what really changes in terms of percent is the amount of REM sleep. But what I want to point out here is that if you consider the total amount, even when we are 80 or 90-year-old, in healthy elderly, still people report sleeping 6, 7, 8 hours. So across the entire lifespan is really around a third of our time. Now, this may be one indication that indeed sleep is somehow important. We spend so much time doing that. Another indication is that if you try to postpone sleep, the pressure to go to sleep becomes very quickly irresistible. And you may end up, it may have happened to you, to be found in strange positions, very awkward situations, because the pressure to sleep was so irresistible. So that's another indication. The third one, and I think this is one of the most important, the strongest evidence that we have that sleep is important, is that it's a universal phenomenon. So of course it's not only us, humans, that sleep, but it's safe to say that all the animal species that have been carefully studied so far do sleep. And of course that means not just the mammals, including marine mammals, invertebrates. We will see about the fruit fly, Drosophila. On top of the slide, you see some of the latest additions to this list. For instance, on the left, that's the sea snail, Aplysia californica. And on the right, you see the flatworm and Cassiopeia, which is a jellyfish. And Cassiopeia is actually, to my knowledge, the only example so far of a brain that is not centralized, is actually a network, and yet Cassiopeia meets all the criteria that we are using to define sleep that I now will explain. Now, when I say sleep is universal, I still have to qualify the sentence because there are more than 1.2 million of animal species, and we have studied sleep so far in less than 300 of them. So it's still a very restricted view. It's also very skewed because most species are mammals, but still I can say that so far the evidence is that all animal species carefully studied do sleep. And so what are the criteria that we're using? This is the one that apply to flies, but actually these are the ones that apply to all species that have been studied. And so the demonstration that fruit flies do sleep came in 2000. It was our lab and another lab in UPenn, led by John Hendricks and Amita Segal. And you have learned at the beginning of this week how flies are so crucial to understand the circadian rhythm and the molecular clock. And that's why we thought that it could be very important also to study the basis, the molecular basis of sleep. And that's why we study whether their behavior meets the criteria for sleep. Now, the single most important criteria is the one that you see on top right, increased arousal threshold, which means that when we are asleep, partially we lose the ability to respond to a stimulus. The second very important factor is on the second row is the regulation. There is a very complicated system in the brain that is telling us when and how to initiate sleep and to maintain sleep. And that's also another indication, of course, that sleep must be an important process, because if you instead buy the null hypothesis that sleep is just a time filler, you would imagine that there is no process to regulate, that there is no process that helps you to recover some of the sleep that is lost in case of sleep deprivation. Now, you may notice that I didn't really push the immobility aspect. It's true that the great majority of species are immobile when they are asleep, more or less, but it's not a requirement. Marine mammals do move when they are asleep. Now, there are many other similarities that are not crucial for the definition, but still we see these similarities. And I must say, for many sleep researchers who were trained, like me, studying sleep in people or in humans or in cats, some of these similarities there are almost troublesome because there are so many similarities, including inter-individual variabilities. Some flies can be short sleepers, others are long sleepers. The sleep changes with age. If you give flies caffeine, they stay awake, for instance. And very importantly, and I will come back to that, if you sleep deprive flies of sleep for one night, you will see impairment in learning and memory and performance. So I want to go back to what is really the essence of the definition of sleep and what we look for to define sleep in, let's say, in a new animal species. Increased arousal threshold means partial sensory disconnection. As shown in this beautiful painting, potentially when we are asleep, we are in a very dangerous situation. So if you think about that and what I told you, that all animals do sleep, and again, we spend a third of our life, we reason that there must be at least one very important function that sleep does that can only be done during sleep and requires this aspect of the brain being offline, being partially disconnected from the environment for that function to occur. Otherwise, it would make much more sense to do the same function during wake. It would be much less dangerous if it were possible. So evidently it's not possible, but why is that? And why do we need sensory disconnection? And I would argue that we need to produce this process of synaptic homeostasis, of regulation of the weight of synapses that are the way neurons talk to each other. That's the hypothesis that we are testing. I was yesterday at the museum here, wonderful museum at Carlston. I found out for the first time about the description of the 6 necessities that were recognized already in ancient times that are needed for our well-being. And it's very telling that at that time they already realized they are listing body rest and sleep as 2 separate entities, as it should be. So it's very important to remember, rest is not sleep. Immobility, quiescence is not sleep. For being asleep, we need to disconnect from the environment. And so if you run the marathon and your muscles are fatigued, you can recover that fatigue from just laying down on a sofa and watching TV, but still being in quiet wake, but still awake, and so able to respond. But your brain will not recover. For that, you need the sleep. And so that's the fundamental distinction. Now, in one sense, you can say, well, we all know why we sleep, correct? We all know that if we are sleep deprived, everything is terrible. It's difficult, it requires much more energy, everything looks dark and blue. And indeed it's true. It's everybody's experience. The question is, why is that? What is exactly in the brain that needs to be restored and cannot be restored during wake? Now, the evidence that is really the brain that needs sleep first and foremost comes from many studies showing that if you don't sleep, brain function, cognition in general, is extremely impaired. And it's hard actually to find a single example of a cognitive function that is not affected by sleep deprivation. So it's vigilance, attention, especially sustained attention, so the ability to keep being attentive to a stimulus, especially if it's boring and long-lasting, ability to learn, ability to consolidate what you learned yesterday, so memory consolidation, ability to integrate a lot of different information, or speak clearly, and what are considered very high-level functions, for instance, risk assessment or humor appreciation. Those abilities, they all are impaired even by one night of sleep deprivation. So I told you, I think, several reasons why we think sleep is important, is universal. Without sleep, there is huge cognitive impairment. And I go back to this issue that is a regulated process. Again, so if you are sleep deprived, there is a system in the brain that is helping you to recover at least some of the sleep that was lost. And again, going back to the topic of this program, that is time, there are actually two systems in our brain. One is on the left, the clock, the circadian system. That's the system that you have learned in humans, is located in a very small region of the brain that is called the suprachiasmatic nucleus, that is literally keeping track of the 24 hours. And so in us, like in flies, diurnal animals, is telling us that the best time to go to sleep is the night and not the day. But then there is another system on the right, which is the homeostatic system. That is the one that is keeping track, not of the 24 light-dark cycle, but of how long you have been awake. It's the one that then is telling you after 16 hours, 17 hours, you better go to sleep. The pressure to go to sleep starts mounting. This homeostatic system is much less characterized. It's probably a diffuse system, so there is not just one single brain area that is somehow getting this signal. And usually the two systems work together so that they potentiate each other, and so that we go to sleep at night and our sleep is long and restorative. But there are, of course, occasions like I am now in jet lag, in which the two do not agree, and then there is a conflict. Now, going back to the question, why do we need to sleep? Why spending 1/3 of our life asleep? That's our riddle. And over the last 30 years, we are trying to address this question in the lab. And it's a difficult question, and we have always tried to address it by using many different models. So literally from flies to rodents to people to computer simulations. Again, starting though from the assumption that the sleep must have at least one fundamental function that is so basic that is shared across species. So if I find something that is only true in us humans but doesn't apply to a mouse, to me, very likely that's not a fundamental essential function of sleep. So first of all, I want to acknowledge that all the work that I'm going to tell you about is done in collaboration with my long-term collaborator, Giulio Tononi, at the Wisconsin Institute for Sleep and Consciousness Research. And the work is all funded by NIMH and NDS in the United States. So the idea that we have been testing over the last 20 years is actually very simple. It's called the synaptic homeostasis hypothesis, but the take-home message is that sleep is the price we have to pay, the cost that we have to pay for being able to learn new things every day when we are awake. And it's a very high cost, but of course something that we are all willing to pay. So it's related to synapses, to synaptic homeostasis. What do I mean by that? I know that many of you are not neuroscientists, so I just go through these basics of how the brain works. So the major cells in the brains are neurons. You see here 2 of them on top, one neuron, an input neuron with the cell body, and then there is this long cable, the axon, through which this neuron is contacting a second neuron, the target neuron. And usually this contact happens in that regions where there are dendrites, protrusions of the second neurons, and that's where the contact happens. And the signal gets transmitted from the input neuron to the target neuron. And this point of contact is the synapse, is the synaptic connection. And synapses are really the bread and butter of how the brain works. Most synapses, 80% or so of the synapses in our brain look like this. They are excitatory. There is an excitatory signal coming from the neuron A to the neuron B. And the pre-synapse that you see, the axonal button, is an enlargement of this cable. And the green dots that you see is where this signal, this chemical signal, glutamate in this case, is contained and then released to affect the second, the post-synapse, so the target neuron, in this enlargement of the dendrite that is called a spine. So that's the structure of how neuron A and neuron B can establish physically a contact between the two of them. Now, the fundamental point that I made before is that when we learn, and we learn all the time, in the great majority of cases, learning implies, leads to a strengthening of these connections. So the signal between neurons that are involved in learning gets stronger. It's not 100% of times that this is the case, but let's say the great majority of forms of learnings. And this is, skill learning, learning a new sport, a new movement, exploration, ability to recognize new patterns, so visual, textual discrimination, fear conditioning, all these forms of learnings end up producing a strengthening, a potentiation of the connections among neurons. And the other very important point that I think if you have to remember one thing from my lecture is this one, that you have to remember is not that we learn only, hopefully, when you come to a lecture. You are learning all the time, because learning by definition means that your neurons, your brain is adapting to the environment, and the environment is always changing. So plasticity, modifications of connections is happening all the time when we are exposed to the environment, so during our waking day. So yesterday for me was the first time here. I have all these traces of learning in my brain related to the wonderful experiences. And you can do the same if you tonight try to remember everything that you did today. And you will be surprised actually by how many things you are remembering. I'm not talking only, of course, about the lectures. Try to remember what you had for breakfast, exactly how you got here, etc., etc. Again, you will be surprised. Many of these traces of learning in the long run are totally relevant, and we better forget them. But in the short run, the brain is extremely capable in laying down all these memories. So, going back to this idea, the left side is the day, the waking day, 16 hours, 17 hours in most of us. During that period, overall, because we are learning all the time, many of the connections, the gray lines in our brain, all these connections among neurons are getting stronger and stronger and stronger. So you end up at the end of the day with many red lines. And if you look closely at the molecular level at the synapses, that is that diagram, the synapses are stronger. For instance, they have a stronger signal that you can record from neuron A to neuron B. Sleep then would be, according to this idea, required to renormalize, to weaken many of these connections in many, many brain areas, probably most circuits. And that's why we see then the green areas, the green lines at the end of the sleep period. So that by the end of the night for us, you again have a leaner brain with weaker connections, but maintaining the memories of what is relevant that was learned that day, and I'll come back to that. And you are ready to learn new things. Now, you can say, well, even if I accept your idea that when we learn, we are potentiating synapses, why can't we just keep going up and up and up and getting stronger and stronger connections? What's the problem with that? Well, the problem is energy. Maintaining synaptic activity, maintaining these connections is extremely expensive from an energetic standpoint. And in fact, there are many calculations. If you look just at the right side of the slide, for instance, there, the calculation is that 34 plus 39%, so 60, 70, 80% of the ATP, the energy in the brain, goes to maintain synaptic activity, and AP stands for action potentials. That is really the signal that is being transmitted across neurons. That's a huge amount of the energy, of the brain's energy budget. And we know the brain is already using a lot of energy. It would just be unsustainable if you were to keep going up and up, even if that was a minute amount every day. But remember, of course, that we are talking about a function that has to apply to our entire lifespan. So that's one problem, energy. The second aspect is supplies, because at the end, these connections, they're very far from, if you remember my diagram at the beginning, the neurons have this long projection, these axons. It takes a complicated process to send all the molecules, the receptors at the end on these connections. And of course, it takes energy. So it's a very complicated process. Third one, saturation. Synapses cannot just get stronger and stronger. And at one point they will stop, which means that you will stop learning. You will be saturated. And in fact, there is a lot of evidence that this can happen quite quickly. You can test formally, force people or a mouse to learn intensively during the day. And then if you try to force again some more learning at the end of the day, it's very difficult, if not impossible. So for all the evidence that we have now, at least for one region of the brain that is the cerebral cortex, actually this issue of saturation is not a theoretical problem. It's happening even within probably 1 or 2 days. So it seems that our brain cells are not very far from the level of saturation. So the process of renormalization is needed very soon. So why? So the idea, I hope you understand, is quite simple. So we learn, we potentiate, and then we need to renormalize. So why is it taking you 20 years or so to test this idea? Well, the problem is that measuring synaptic strength is actually very difficult. It's difficult because there is no single measure of synaptic strength that you can use. There are many different measures, especially if you want to do it in different species and in vivo, so in a freely moving subject. And so that's why it's taking so long. One method that we have been using over the years is a molecular one. So you can use molecular markers to assess the strength of these connections. And again, you go back to this schematic of the synapse, so the pre-component, the axonal button, and the post, the spine head. Both of them have proteins that are very crucial for their functioning, so you can measure those proteins, because in synapses that get stronger, these components, these proteins, are more abundant. And so that's what we have been doing. For instance, you can use a presynaptic protein. This is a fly brain. And on the left is a fly that was able to sleep all night. Flies are diurnal like us, so they sleep during the night. And on the right, a fly that instead was sleep deprived. And red means there is a lot of this protein. This BRP Brook pilot is a marker for synapses in the fly brain, extremely widespread. Basically, every contact has this protein. And so there is much more of this protein after 12 hours of wake relative to 12 hours of sleep. So that's one way you can do it. Another way, very similar, this time in mice and rats, this experiment has been done both, is to measure proteins on the postsynaptic side, on the spine head. And what we measure are these receptors that are really the ones that are assessing how much of this signal of this glutamate is being released. And again, you can extract these receptors from the brain after the animal has been asleep for several hours or awake for several hours and just quantitate their total amount. And if you do so, for instance, you find consistently, and this is now being replicated in different labs, there is 20, 30, 40% more of these proteins after wake than after sleep. So that's another way. Or you can literally, because again, this is the communication across neurons. At the end, how they communicate is through currents. So you can measure an electric signal going from the neuron A, the input neuron, to the target neuron. And if you stimulate neuron A and record the response of neuron B, and of course you keep the stimulation exactly the same, the response, the amplitude of the response will be reflective of how strong their connection is. And so you stimulate A, you record from B, and you can get those kind of response. The bigger the response, and therefore the steeper is the red line, the slope, the stronger is the connection. And so for the same amount of stimulation, you see that the response is bigger after wake than after sleep. And the beauty of this experiment, although again is indirect, you can do this through electrical stimulation in a mouse, in a rat, but you can do this also in people using non-invasively transcranial magnetic stimulation, so a very mild stimulation directly on the scalp, and you can record this kind of responses after sleep and after wake. And the results are very, very consistent. A third method, that is what we are investing now a lot, because we think perhaps is the stringest way of testing this hypothesis, is actually measuring literally and very simply the size of synapses. Because fortunately for us, there is a very strong correlation between structural and functional plasticity. So if synapses get stronger, they also literally get bigger. So the implication of this hypothesis is that literally these connections across a waking day, a few hours of wake, should grow, and vice versa, they should shrink during sleep. Of course, the synapses are small though. Again, the top, the green is the axon, the pre-synapse, and the yellow is the spine, the post-synapse, and the red line that in 3D is actually an area is their area of contact. That is the structural measure of synaptic strength. So to measure this, you need an electron microscope. And that's what we have been doing recently, because, again, to put the thing in context, you know, the largest synapses are around 1 micron. Grains of sand that can be as large as 2,000 microns. Okay, that's why you need an electron microscope. So we have a serial block phase electron microscope that allows you automatically to take a piece, in this case of the mouse brain, and automatically to cut a very thin section and then take an image, and then again another section, take an image, and so build a stack of many of these images. So you have a volume of tissue, in this case the cerebral cortex, in which you can measure these synapses. Now, this process is automatic. The technique now is very well advanced. You can get these stacks overnight. The problem is that to get then the nice coloring, segmentation, and all the measurement, you still have to do this manually. And so that's why it's extremely time-consuming. And so what we do, going back to the schematic, is we need to reconstruct the dendrites, so the trees, the receiving end in these images, and then the axons, and then we need to reconstruct the exact area where the two are touching each other. That's the synapse, and the larger is the area, the stronger is the synapse, okay? This is just to give you a sense. This is just one of those images in 2D. And this kind of snake is a piece of the dendrite, and all the asterisks are synapses, is the post-synapse. And facing them, you see all those dots, so that's the axon. Just to give you a sense how many synapses. The brain is full of synapses, and that's why its synaptic activity is so expensive. Just in the human neocortex, we have 100 trillion of these synapses. So here is how we do the experiment usually. So we have two groups. Of course, you want to see the effect. So you want to take a brain of an animal that has been asleep for many hours, so during the day, because mice, rats are nocturnal animals, and then you compare it, and that's the last group, the S group, that is collected in the afternoon after having slept many hours. And you want to compare it to the top, the animal that was collected during the night after having been spontaneously awake for many hours. That would be the main comparison. But remember, again, this is the topic of this program, time. We know in the brain only, just, but you have learned also about the periphery, there are hundreds of genes and processes that are changing in the brain just because of circadian time, just because it's 3 a.m. rather than 3 p.m. And so when you want to compare and study the effect of sleep and wake, you can't just use those two groups, because you will never be sure that it's really sleep-awake rather than 3 a.m. and 3 p.m. So that's why you need the middle group, the extended wake, the EW. Those are mice that are collected at the same time of day as the sleeping animals, but they are kept awake. We literally play with them. We give them novel objects, so for a few hours it's perfectly fine. They are awake all the time, and so they are in the opposite behavioral state relative to the sleep group, but the same circadian time. So when I tell you about differences between sleep and wake, it's always between the sleep group and the two wake groups, so that we can remove potentially this confounding effect of time of day. And going back, and this is, I think, important for the students to realize, I told you the automatic method, but then you need to do the reconstruction. So this is all the dendrites that we reconstructed for this study that we published last year. And so the total was almost 7,000 synapses that I think is still the world record. And it took 5 years, 5 people working full time to do this. So you need, you better have a good question, and you better have a lot of patience if you want the answer. And the answer is this. So here, each dot is indeed one of these synapses. And the blue dots are the sleep group, and the other two are the waking groups that are incredibly similar, so are indistinguishable actually from a statistical point of view. And what we found, that the size here is the size of this contact, so it's a measure of strength. And what we found is that overall at the population level, synapses after sleep, 6-7 hours of sleep, are 18% smaller than after wake, at least in this region that we studied, that is the cerebral cortex in a very specific layer. And this applies to many synapses, the great majority, at least 80% of the synapses that we tested. So it seems true at the structural level, the most synapses go down in size with sleep. And that's a schematic just to summarize this point. Now, this is published. I want to show you 2 pieces of data that are unpublished with the same method, and again, this took many years. What I've shown you so far refers to this yellow area that is the cortex. It's in the dorsal part of the brain. But we asked, what about the green area, that is the hippocampus, is a deeper area of the brain, very important for learning and memory. And so we did basically the same study in the same animals, and look whether also there we could see these kind of differences. And you see an example there, again, on the left is one of these dendrites in the cortex, on the right in the hippocampus. And you see already a very big difference. There are many, many more spines, many more synapses in the hippocampus, and this was already known. But there is 2-3-fold higher density of synapses in the hippocampus. These are all the pieces that we reconstructed. And the take-home message that I want to give you is that we see the same. So when we compare sleep group with the extended wake group, so killed at the same time of day, again, we see at least an 18-20% decrease in size of synapses with sleep. In fact, in the hippocampus, the difference seems even more dramatic, if you wish, because not only we see shrinkage of the synapses, but in many of them, actually, we see that their number decreases. So there is literally a pruning, an elimination of these synapses with sleep. And the other part that is unpublished is about very young animals, pups. These are only 2-week-old pups. Because you could argue, rightly so, well, maybe this need for the brain, once you have a mature brain with all the connection more or less established, there is very little space, there is very little energy to spare. So if you grow because of learning, then you need to renormalize during sleep. But what about a very young brain that is literally still expanding, forming many of these connections, forming thousands and thousands of these synapses every hour? Maybe those brains do not need sleep to renormalize. We know that they need sleep, they spend a lot of time asleep, but maybe not for this process. And so that's why we study these animals. And here, instead, again, that's the right side, an example of a dendrite. Of course, these animals have fewer spines, synapses, because they're still growing, many of them. And these are all the dendrites that we reconstructed in cortex in these young animals. And the result is actually that the answer is that absolutely not. The process still happens even in a very young brain. Actually, if anything, is even more pronounced. Here, when we compare sleep and wake, the difference on average at the population level is 30%, not 18%. So it seems that this process of synaptic renormalization is also required when the brain is still developing in early age. So this is the general idea about, we learn, we grow literally our synapses, they get stronger, they use a lot of energy, they get close to saturation, that's why we need to renormalize. Now, you can ask, well, maybe the evidence is strong about this imbalance of synaptic strengths between sleep and wake, but how is it happening exactly? Because this process of weakening and renormalization must be smart enough that then at the behavioral level, you can account for all the functions of sleep that I told you about. Meaning, after sleep, we can remember better, we can learn new things, we have also forgotten irrelevant details, and we have been able to do what is called the gist extraction. So we have been able to really distill the knowledge and integrate the new knowledge with the old one. So this process, even if it's happening, it must be smart enough to explain all this. And of course, it will take me 2 more hours to try to explain this. But I can tell you that it is a very exciting time because over the last 1 or 2 years, so it's very, very recent, but there are new data at the electrophysiological and the molecular level that are starting to indeed explain how this could happen when the brain is offline. And basically, the take-home message that you should remember is the following, that is exactly depending on what you do during wake, what kind of neurons and what kind of synapses you are using, that is going to dictate which synapses will get renormalized, so weaken, and which one instead will not, will remain at the same level, and because everybody goes down, actually, relatively speaking, they will be potentiated. And now we have evidence that the specific brain activity that is happening during sleep, the way neurons are firing and they are active during sleep, is very crucial for this process to happen. And as you see on the bottom right, we are starting to identify very specific proteins and receptors and molecules in the brain that are literally tagging the synapses and therefore then allowing these synapses either to be protected to escape the process of renormalization or instead to undergo this process of weakening. Thank you.
Thank you very much. So, ready for questions? Over there, who is helping with the microphone? Volunteers, there you are. We need the WEP volunteers on the ground. Thank you. So there's a gentleman over there. Please. Raise your hand. No, there was... Okay.
The second row.
This gentleman first. Sorry, we have to respect an order, otherwise... Thank you very much. Good. Let's get started.
Thank you. Thank you for the very nice presentation. So you focus a lot on learning during the day and then the renormalization to allow more learning the next day. But if you see it more as data collection during the day and then having more selective renormalization that actually is the learning process, is that, according to your hypothesis, would that be contradicting your hypothesis? Have you thought about thinking about it in this way?
No, you're perfectly right. I mean, you can call it, we call it a smart down selection. And so if you apply, so if you think this is a form of learning, certainly, but it's based on what happened during wake. Indeed, there is a process of selection. If it were the case, and again, I didn't have time at the end to go into this, but if it were the case that all synapses would be downscale and in the same way, and all of them, you would not maintain any form of learning. So it has to be selective, definitely. Yeah, it's a question of wording. Yeah.
Yeah, thank you very much for the interesting lecture. I got two questions. The first one, where does the sleeping exactly happening? Is it in the brain, in the nerve system? And a following up question, do viruses, bacteria, plants sleep? And thank you.
Okay, the virus, I don't know. We assume, so far, all the animal species in which sleep has been defined as some kind of complexity of at least the neural networks, as I mentioned, the Cassiopeia, rather than a centralized brain, but it's still a very complicated network, groups of neurons. The brain sleeps, meaning in the brain, there is a fundamental difference in the way the brain works between sleep and wake. And it's not, as it was thought a century ago, the neurons stop firing, stop being active, they just shut off during sleep. That's absolutely not the case. In fact, overall, the level of activity is only maybe 10-15% lower overall in sleep than in wake. But it's the pattern, is the type of activity that is very different, and that's what I mentioned at the end, and that type is probably what then allows the renormalization. I don't know when you say the neural, the brain, for me the brain, I don't know if you wanted to pinpoint the difference between centralized brain and peripheral brain. From the centralized brain, of course, then there are effects also on the periphery, and so there are differences in many processes driven by sleep, but many of them can be also driven by rest. Instead, the difference between activity for the brain is absolutely specific for sleep and not for rest.
Over here.
I have two questions. I'm curious. I had my coffee after lunch before the talk, but I guess some people didn't. What's the effect of coffee or caffeine or other stimulants in sleeping? And my second question is, are there really differences in sleeping physiology between males and females?
So the second first, there are differences in mammals, in humans. They are very, very subtle. In some species, like fruit flies, they are a little bit more pronounced. So, for instance, male flies tend to sleep much more also during the day. They have a kind of siesta that female flies do not have. And these differences are really mainly driven by light. So flies are extremely sensitive to light, more than we are. But the differences, again, there are differences, but there are minute differences. They're not fundamental differences, especially in terms of also of how sleep is regulated. Now, your question about the caffeine, so you're saying if I drank a caffeine that keeps me awake. That certainly. The caffeine is the most, still remains, actually. If you take the right dose of caffeine is as potent as any of the most powerful amphetamines or drugs that you can take. It's the most, it's just a very high dose. And the way it works is that it blocks one system that is the adenosine system. And the adenosine is a molecule that builds up in the brain as a byproduct of energy metabolism. And so it's one of the signals that is telling us, well, you have been awake, you have used a lot of energy, adenosine builds up, adenosine tends to blocks these synapses. And so by taking caffeine for a while at least, you are trying to remove that signal that is, forcing neurons to fire less.
Hello? Yeah, what about dreams? Is it like a consequence of this or like a whole different process completely?
It's very difficult. It's probably, well, I don't know. It's very difficult, if not impossible right now to answer because, to make a connection, you will have to study dreams in animals. And still, dream research, even in humans, is all based on verbal report. I'm saying still because I think I'm very hopeful that soon we will have neural correlates very well defined so that you can, be pretty sure that the subject was dreaming or not, even without waking up the subject and asking what was going on. But right now we are not there. And I'm not sure. I think I would bet, but that's my gut feeling, that they are not related. But, I have no evidence for that.
Yeah, but series of questions. People in the back are simply stretching or they want to ask questions? Okay, 1, 2, 3, good. Then there's one over here. There you are. And one. You first, yes, and then in the back.
Does brain death considered as sleep?
Brain death?
Yeah, people who die with unconscious.
No, it's a state further. The definition of sleep is a reversible state. So by definition, when you are asleep, yes, you are less able to respond to a stimulus. But if the stimulus is strong enough, the noise is strong enough, you will always wake up from sleep. And that does not apply to brain death, to coma, to anesthesia. That's the way, actually, you can distinguish. So sleep is not brain death, no.
In the back. Wait.
Hello? Yep, okay, cool. First off, thank you for your talk. That was really interesting. My question is, what are some of the other hypotheses of why we sleep? And I'm curious how your hypothesis kind of jives in or around those other ones, if it does.
Thank you. There is many ideas about sleep functions are related to this issue of plasticity, synaptic plasticity, and learning and memory. So from that point of view, there is big agreement in the community. What we disagree upon is the mechanism to obtain, to favor synaptic plasticity through sleep. So our idea is this, that you get there through synaptic weakening. Other hypotheses think that actually you learn during wake by potentiated synapses, and you keep doing the same also during sleep. And so even if you are offline, those synapses keep being potentiated. Why we think this is not happening? Because to potentiate synapse, you need the condition that is typical of wake, in which many molecules, many neurotransmitters in the brain are all conducive to synaptic potentiation, but those are not present during sleep. So the end result is, as I alluded to, indeed a relative increase in synaptic strength after sleep of the synapses that you used to learn, but is relative, is simply because everybody went down, not because these ones that were potentiated during wake keep going up. You see, that's the main contention, I think, right now. And I mentioned at the interview this morning, I can see why in a few years we will be able to address this fundamental difference, because finally we are able to follow in a living animal synapses in vivo, the same synapse, and study what happens to the synapse after learning, before learning, after learning, and then after wake and after sleep. And then we will know if really there was an absolute change in synaptic strength or only a relative change.
So we have a flurry of questions. I think Laura first.
Yes. So first, thanks for coming to Chaos for this nice talk. My question is, so following this theory, but do you hear me anyway? No? No, okay.
No, speak closer to things.
Okay, so following this theory, after waking up, your brain is much more active and ready to learn. And this is actually, I think I'm a good example of these things. My brain in early morning is very active. I can work and learn much better. But I think this is not the case for everyone. There are many people that start to be active after many hours after waking up, and they are actually very active during night. How do you explain this diversity?
Yeah, so there is a phenomenon that is called the sleep inertia that I didn't mention. This is happening actually to all of us. So when you say, and that's why when we do experiments, we wake up subjects, we actually then wait at least usually half an hour, because for most of us it's half an hour in which you have literally, we have shown and other people have shown, some of your neurons are still asleep. So some of your neurons, not the entire brain, there is enough of your brain awake that you are awake as a whole. But some of these neurons still have a pattern of firing that is much more similar to the sleep one than to the wake one. This sleep inertia, as many of the sleep phenomena, has huge inter-individual variability. So some subjects are ready to go maybe in 10 minutes, but basically nobody is ready after 1 minute. On average, it's 20, 30 minutes. That's why we wait 20, 30 minutes. Some people have terrible sleep inertia, so much so that many of them, even if they recognize, for instance, that they could, in the afternoon, take a nap and it would be very restorative, the sleep inertia is so long that, after a nap, then it takes them until 7 or 8 p.m. to really wake up, so they don't nap. So that's one thing. The other that you mentioned, again, I mentioned that there are two mechanisms of regulation. One is the clock. Some of us wake up at 6 a.m., they are ready to go, and we prefer to go to sleep at 9 p.m. Because the clock, the suprachiasmatic nucleus, is telling us the tau, the period, is less than 24 hours. Other people have a period that is longer than 24 hours, and they tend to be the late people. That's due to the clock. But once you adjust that, then the period in which you work and you function is similar. It's just that it shifted across the 24 hours. And again, there is huge variability. In the same way, the homeostatic process is different. So some people really need, 8 hours, 9 hours, 10 hours. Other people need much less. There is big differences.
Good. So we have one question from the back, one question from the front. over question over there.
Thank you for the talk. So you kind of partially answered my question actually just now, but I'm more interested on the microscopic level. So sleep inertia. Initially, my question was, is napping enough time, like midday naps, are they enough for pruning to occur? And sleeping inertia, what is it in terms of on a micro scale? What's the mechanism?
Well, the mechanism we don't know, but there is more and more evidence that, again, the brain doesn't wake up all simultaneously. The same way, actually, we know that it doesn't go down to sleep. All the brain areas don't go exactly simultaneously. So some part deep, the thalamus, usually goes to sleep even 60 seconds or 1 minute, 2 minutes before other parts. And now that we have the ability, because there is a lot of sleep research now done with deep recordings in people who are implanted with deep electrodes because of epilepsy. So we have this data anyhow. Of course, you don't implant deep electrodes in humans unless it's for clinical reasons. But now because of that, we know. So, there is this process that is very complicated that really tries to put the brain asleep all at once and wake you up all at once. But it works okay, it's not perfect. So that's one. The second question I forgot. The sleep inertia is this variability.
More mechanistic aspects.
Why is that? It might be due to the fact that, to go to sleep or to wake up, you need systems, the so-called arousal systems, so projections from other areas that have a strong waking signal or sleeping signals. And the way these areas project to the rest of the brain varies. So some of the signals are stronger in some areas than others. So that's probably the mechanism.
Okay. Here and over there.
Hello. Thank you again for the informative presentation. I had a question about actually the correlation between sleep at night and the brain recovery or the synaptic strength. So is there a correlation between them, a relation or?
You mean in terms of duration or?
Or no, actually, brain recovery and sleeping at night. Like, do you recover better when you sleep at night?
Relative to the day. Yeah, the second question was the nap. Now I remember. Well, we don't know yet. So we don't know how short sleep can be and still provide you this restorative power in terms of, for instance, renormalization. Okay, so far, because it takes so long, it's so difficult, so far we have really considered the bulk of sleep in a mouse would be 6-7 hours versus most of the wake period. Whether maybe only 3-4 hours rather than 7 are enough, whether a nap is enough, we really don't know. My bet again would be that for having the full restorative power, which requires these structural changes, literally structural changes, is not going to be seconds or minutes, it's probably hours. So you can recover a little bit, and we all know probably by a nap, you can wake up refreshed, but maybe just it buys you a little time. But it's not that you can then go for the entire 16 more hours. Okay, but there are very few studies testing that, how little you can go and have the full power. There is a lot of research being done on that issue, and especially on the fact that we realize that what counts probably is not the duration, is the quality of sleep, so the depth of sleep. And one thing that now is very obvious is that you can increase the quality of sleep by acoustic stimulation, which seems paradoxical. Of course, if the noise is very strong, you wake up. But if the noise is very subtle, actually the noise will entrain brain activity and these big slow waves in the brain that are so important for the renormalization. So the way now people are trying to do power sleep is not by using drugs, is by using this acoustic stimulation that seems to work. It's very tricky though, because of course he has to be monitored. Every subject is different, but it seems to work.
So one last question, so over there.
Hello. Thank you. My question is how the consolidation of memory work during sleep time?
How the consolidation, that's a very difficult question to answer. The consolidation of specific memories probably, it happens because those memories are protected from the renormalization, so they don't go down. But the consolidation very often, at least for what we know now in animal studies, very often doesn't happen in just 24 hours. And we are going back to this issue of time. The real consolidation of memories for many memories is across days or weeks or years. So to really answer your question, we'll have to really follow the process for a long period of time. And we sleep researchers usually, look at the wake and then sleep and that's it. But we have to start, and people have started doing that, to follow the process for much longer.
Okay, let me thank you, first of all, the audience for having this very exciting
thank you.
Conversation with you and
very
yourself.
Happy. Thank you
For the great talk. It is my privilege also to offer you, on behalf of CAUS, this
wonderful.
An astrolab.
Oh, thank you. I saw it in the museum yesterday.
Yeah, it's a contribution of course, the Islamic culture today,
yes.
But also perfectly fitting the time.
Yes, thank you. Thank you very much.
Chiara, thank you very much.
Thank you.